Hello, everyone. Thank you guys all for joining in online, and thank you to our guests for coming through. So this is our pharmacogenomic awareness event, and we are partnered to do this with Thermo Fisher. Firstly, some house rules. Please feel free to grab some drinks, some snacks, and the bathrooms are just to your right. So my name is Kayla. I am the product manager for PharmaGene, which is our pharmacogenomic test. And this is what we are going to be talking about today. But I just wanted to give you a little bit of background as to who Next Biosciences is. So we've got 4 divisions to our company. First is our Next Biobank. So we are actually a cord blood bank. So that means that we store the cord blood and the stem cells from a baby's umbilical cord. And that can basically be used as a medical insurance for the future if the baby ever needed a bone marrow transplant or something like that. Next, we've got our Next Genetics. This is our genetic test that we do in the fertility space. So, for example, our non-invasive prenatal testing or our products of conception. Then we've got our Next Biologics. We actually use the amniotic membrane of the placenta, and we make biological products from this. The healing powers of the membrane of the placenta is incredible. So we can make products that are great for wound healing, can be used in ophthalmology and orthodontistry. And finally, we've got our Next Health division. This is where our pharmacogenomics test falls under, and we've got 2 other tests that fall under this space called Biome and 3x4. And I'll get into that a little bit more now. So the idea behind our Next Longevity Health division is to extend a person's health span, because with all of the medical advancements, our lifespans are extending, but what would be the point of that if our quality of life wasn't great towards the end anyways? So firstly, we've got our Biome test. This is our gut microbiome test. So we test for the bacteria and fungi that are currently present in your gut. And we use that data to create insights for a person to improve their gut health. So we'll give you food recommendations, supplement recommendations, and this is all around to improve your gut, your metabolism, your gut-brain axis, etc. Then we've got our 3x4 nutrigenomics test. With this test, we are testing for 36 different pathways that include your cardiovascular systems, what exercise would be best for you, your caffeine tolerance, and it's all about creating a health plan and living better according to your own genetics. And then we've got our PharmaGene test, which is our pharmacogenomics test, which is what we are going to be focusing on today. Basically, what pharmacogenomics is we're looking at the role of the genome in medication response. And this is essentially to optimize every treatment's patients according to their own genes. So we've got 3 awesome speakers that are going to be telling us a lot more about this today. We've got our Dr. Marieke Coenen, who is going to be representing Thermo Fisher. She's going to be touching on the technicalities behind the pharmacogenomics. So she's going to be talking about the arrays and everything that goes behind the genotyping and processing. And then we've got Dr. Jeff Schaman. He is the Chief Science Officer at Coriell Life Sciences, and he's going to be talking about the science behind the bioinformatics from Coriell, and this is relevant because we are using their bioinformatics to supply that here in South Africa. And then finally, we've got our Dr. Andrew Macleod, who's actually here with us today. And he is a pharmacogenomic expert, but he's also brilliant to talk about this because he's got 28 years of experience in a clinical setting. So he's going to be making this pharmacogenomic data relevant to our healthcare practitioners and really putting it into the context of a clinical aspect. So I'm going to transfer over to our first speaker. This will be Dr. Marieke Coenen. Sorry, I just have to. Okay, so Dr. Marieke Coenen, you are now.
I'm
host.
So can you see the slides, just to make sure? Okay, perfect. So today I will show you how we use the Thermo Fisher methods for our pharmacogenetic testing in clinical practice. So as I said, I will show you the methods of Thermo Fisher Scientific, but I will show my own ideas and own thoughts of how to use them and how we implement them in our routine practice. Just to start off with, what are we aiming to do with pharmacogenetics? We are looking to genetic variants that influence the individual response on medication. And when you give a patient medication, it needs to be absorbed, distributed, and metabolized in your body. And in all these aspects, genes play a role, so they can have an influence on that. And that, in the end, will make if a patient will give a positive effect on the medication or maybe will develop side effects. And we are looking to many genes, but that are mainly genes that are involved in the metabolism of the medicines. So this is the set of genes that we analyze at our center in the Netherlands. So these are about 20 genes, and we test them weekly for pharmacogenetic testing. So we have one gene, the CYP2C19 gene, that we test daily, and this we do specifically for clopidogrel. And we have 3 genes that we test twice a week, and these are mainly for cancer treatment. So pharmacogenetics testing at Erasmus MC started in 2005, and we have seen a clear increase in the number of tests that are requested yearly. So in 2016, we had about 7,000 tests. In 2018, 14,000. And this year we will end with 30,000 tests for pharmacogenetics. So in our routine testing in our center, we use 2 methods, the autogenomics Infinity, and we use several methods from applied biosystems. So these are mainly the open array and specific assays for calling copy number variants. So we test all samples with both methods, and the results should match. And if the results match, then we make a report for the patient and the doctor. If not, we repeat genotyping, or we use a third method to make sure that we give the right results. So once we have the results, we create a report for the doctors, and this is an example. It's in Dutch, but probably in South Africa you can read a little bit of this. So we give the results of the genotyping. So we report the star alleles because that is the genotype results, and we also translate it to a metabolism status. So we say, for instance, in this case, CYP2C19 metabolism is ultra fast, and the CYP2D6 metabolism is slow. Besides this report, which we send out to the doctors, we also create a card with a pharmacogenetic profile for the patients that they can show to all their doctors, physicians, but also other people that need this information. And besides this, we also have an app that can be used by the patients, which gives some basic information about the medicines and how the genes are involved in processing of these medicines. And within this app, their own specific pharmacogenetic profile is presented as well. So now to the more technical and the methods. So the pharmacogenetic assays that we use from Thermo Fisher Scientific are single assays using Taqman. So these assays specifically target one variant, and you get a result about the genotype of that specific variant in a specific gene. And we also use the Taqman open array, where you get results of many variants and many genes in one go. For this, you have the 60 panel and the 120 panel. And for some research projects, we also use the exome. So first, I will show you the tests that we use in routine testing for pharmacogenetics. So these are the Taqman single assays and the open array. And actually, the majority of our routine testing is based on this open array together with the other method that we use. So for the open array 60 panel that we are using at the moment, that covers quite some genes that we do in routine clinical testing. So this is again the slide that I showed you before with all the genes that we do in our center. And if you overlay this with the genes that are present on this open array, then you see that about half of these genes are covered, but actually the genes that we do most often for the patients are covered. So the genes that are not covered at the moment, we do not test weekly because they are not requested that often, except for the oncology genes, but those we do with different tests anyway. On this array, there are also 2 other genes present, but those we do not test in our routine testing. So the very nice thing about this open array is that you get results very fast. So this is the workflow that you do with this open array. You get your sample, so your blood or your buccal swab. In our case, we do not use saliva. Then we isolate the DNA and we prepare the sample, and this can be done in less than 2 hours. And once you have your sample and your mix prepared, you transfer everything to the sample loading system, so the AccuFill system, and there your sample and your mixes are prepared to do in the end the real-time PCR on the Quant Studio. And this takes about 4 hours, and then you have your analysis and your report, which takes about 30 minutes. So you can do this easily within 1 day. And if you want, you can run up to 1100 samples daily. So with this assay, you can test for SNPs, but you have to do the copy number variance, which is highly relevant for the CYP2D6 gene, because this gene can be deleted or duplicated separately. And that is what we do using the Taqman CNV assays. And this is just an overview of a result of tests that we do weekly. And what you see here on the x-axis are all separate samples. On the y-axis, you see the copy number. Intron 2 is indicated in green, and exon 9 is indicated in blue. And what you see in the first part of the figure is that many samples have 2 copies of CYP2D6. So that is the normal situation. But we also have some samples that show that they have 3 copies in intron 2, for instance, and 2 copies in exon 9. And these are the more difficult CYP2D6 samples. So this we combine with the SNP results of the open array. And in this case, the patient was homozygous for STAR10. And if we then combine these results, then we get our final result for CYP2D6. And that is in this case that the patient is homozygous for STAR10 on CYP2D6. But in addition, this patient carries a hybrid of CYP2D6 combined with CYP2D7. And that we know because there are 3 copies present of the CNV in intron 2 of CYP2D6 and 2 copies of exon 9. So we know that there is a deletion at the end of the gene, and that is always combined with a CYP2D6. So that is the hybrid that we can find. We also use these assays for other difficult samples, but due to this time that I have, I cannot show you that now. So I will now move to the new open array. So this is the Taqman Open Array 120 panel, and we had early access to this panel. And what happened in this assay is that the target list was expanded, and also some difficult, some variants that were difficult to detect with the 60 panel were now better in this panel. And what brings that for us in clinical practice? Actually quite a lot. So I will go back again to the slide with the genes that we normally do for our routine testing. In bold, you see the genes that were also present on the 60 panel. And actually in this 120 panel, there are more variants in these genes present. So we can do better genotyping for these genes. But in addition, other genes are also now in this 120 panel. So actually, we can do less of our single Taqman probes if we use this panel. So these are the genes that we test in our routine, but there are also some more genes present on this 120 panel. So I cannot show you a lot of results, but we did some initial testing of these genes and variants, and it looks very promising. So we are aiming to move to this more extended panel. And as a last example, I want to show you the pharmacofocus array. And this is actually an array that we now only used in research setting, but the nice thing is it's fully configurable. So if you want, you can add your own variants and your own genes. What is also very good is that you can do SNP genotyping in combination with copy number variants, which is not possible on the other method that we use. And actually, even though you can test a lot, much more than on the 120 panel that I showed you, you can still do this in a 3-day workflow. So you have your reagent, you prepare your target DNA, and then you run it on a gene titan machine and you get your analysis. And within this analysis, star allele reporting is included. So in total, you cover 66 gene regions, but you get star allele calling for all relevant pharmacogenes, which is very helpful. So we did some research analysis with this approach, which is pharmacofocus array. And actually what we saw, and I'm just showing you a few genes, is that for blood, we had a very good reproducibility in 10 samples. But for the buccal swabs, which we also often use, reproducibility was much lower. So we need to optimize a little bit for this. But for the precision, again, for blood, we used 22 samples. It was very good. And for buccal swabs, again, it was a little bit less. So in conclusion, the Tocman Open Array fits very well in our routine flow. It's fast. We have a turnaround time from sample preparation to report of 5 days with this. The little bit of downside is that we have to do the CNV separately, but still it's very easy to do that together. And we will move to the 120 panel because this will make our life much easier because we can do many more genes that we are interested in one go. We use the Tocman assays for complex samples. I gave you one example, but we also use this for more difficult questions, and that is also very helpful for us to give the good results for CYP2D6. And finally, for the pharmacofocus array, we have very good results with blood. The buccal swabs we need to optimize. We can use it for complex samples, for instance, for CYP2D6. We can run 94 samples in one analysis, and you can do the CNVs and the SNPs together. And finally, I would like to thank our pharmacogenetics group at Erasmus MC, Ron van Schaik, Maya Matis, and Gemma Brufau, who did a lot of these testing, and all technicians that were involved in this analysis. And I would like to thank you for your attention.
Thank you so much, Dr. Coenen. That was really interesting. Thank you for that.
Shall I stop sharing just to?
Yeah, you can.
Yeah.
You can stop.
Okay.
Next, we are going to have Dr. Schaman as our speaker. I'm just transferring the screen over to him. Here we go. Thank you.
Good afternoon, everybody. Kayla, can you see my slides?
Yes, I can. Thank you.
Fantastic. All right, I'm going to continue on where our last speaker, Dr. Coenen, kind of left off. The technical pieces are extremely important because that is the data that we're going to combine with the information on drugs to get to our physicians and our patients. So just briefly, Coriell Life Sciences was founded 10 years ago out of the Coriell Institute for Medical Research, which is in Camden, New Jersey. We're actually located in Philadelphia, in Pennsylvania on the East Coast. And what we're going to talk about briefly today is that, something that we all understand is medications don't work for everybody, right? We've all had this happen either to ourselves, a family member, a grandparent, a friend, that they've taken a medication and it just hasn't done anything. The other option there is that they've taken a medication and they've gotten an inappropriate side effect, something that makes them not want to take that medication, right? And these are the things that we want to avoid. And the only reason we're having this conversation today is because we can preemptively test genetics to avoid those efficacy and safety concerns. That's why we're having this talk. I'm going to talk a bit about how this works. And a program that we've put together, putting pharmacists at the center of care so that those pharmacists can review genetic and also non-genetic information to get back to the physicians and then to their patients to make actionable changes. I'm going to give you a little bit of background on some of those studies and the outcomes of those in the short time that we have today. So this is information from the United States, but what I've learned working all over the world is that this isn't isolated to one area. If you're on a medication, the chances for emergency department visits due to adverse reactions themselves, and even things as severe as fatalities due to these is higher the more medications that a person is taking. So removing that is of high importance to all of us. A couple of the examples that I'm going to use today are treating depression, major depressive disorders, and also some of the cardiology challenges that we have in the clinic. But what I also want everybody to know is that this technology can be used in any patient, even for patients that are not currently on a medication. So let's start with depression really quickly here. We know that a lot of these antidepressants take 6 to 8 weeks, even 12 weeks to start working in our patients. That's just the biology of these medications. They need to build up in the synapses to be able to start working. Once that starts working, or once that 6 to 8 weeks are up, about 50% of patients do not actually respond to a medication. Those that do, about 30% discontinue treatment due to intolerable side effects like weight gain, sexual dysfunction, and other things. And we know that there's non-adherence, those people who just won't take the medication because of some of these reasons. The reason we're talking about pharmacogenomics is we know so much about the pathways of metabolism for these medications that we can look at the genetics, and before a patient even starts taking these drugs, we can tell them if they're going to work or not. We can tell them if they're going to have side effects. So we still need to wait 6 to 8 weeks to see those things working, but that trial and error prescribing, which is standard of care right now, is something that we can avoid as well. So I think everybody in the room hopefully understands a little bit about pharmacogenomics, but basically what happens is we have a genetic variant that changes the molecular activity. And what that does is changes the metabolism, the pharmacokinetics or pharmacodynamics within that patient. And that induces the effect. So this is ADME drugs, right? We're talking pharmacokinetics, pharmacodynamics, right? The absorption, distribution, metabolism, and elimination of medications. And that includes both the elimination, but also getting medications across the membranes, right? Even if it's metabolized correctly, if you're not getting those medications to the right place, it doesn't matter how well that drug is should work. What we have is the common scenario that's in most drug labels, right? Take 2 of these and call me in the morning. But that doesn't work for everybody. So Dr. Coenen mentioned that there are these ultra-rapid metabolizer, and CYP2D6 is a fantastic example of that. And what happens there is you've got duplications of this enzyme, of the genetics that causes this enzyme. And we want to make sure that we're testing for that, for one, which we all do, and that we have those medications and understand the outcomes for them for those ultra-rapid metabolizers. We also have the issues where there are genetic variants which causes a slower metabolizer or slower metabolism of these medications, causing those drugs to either build up in the body, or in the case of a prodrug that needs to be turned on, those drugs are unable to be turned on. And so what we have is the same diagnosis and the same prescription, but in different patients, we get different outcomes. With genetics and pharmacogenomics in particular, we can avoid some of that. Now, this is available and useful in all sorts of therapeutic areas. But again, even though the oncology pie wedge here is the largest, we know that nobody is just dealing with oncology issues, right? There are pain issues. There's nausea and vomiting. There's depression. So neurology, psychology, anesthesiology, gastroenterology, all of these subspecialties are able to be using pharmacogenomics to help out. I'm not going to spend any time on this because you learned a little bit about this in the previous conversation, but one of the reasons that we talked a lot in that first discussion about 2D6 is that it is associated with a lot of medications. And we've known about genetics for quite a while. So G6PD actually is implicated in Favaism, which Pythagoras, you might remember the story, he was being chased by some soldiers. He was run into a fava field and he would go no further because he knew that fava beans would react poorly with some people. And that's actually because of G6PD, which is implicated in a lot of our anti-neoplastic medications. The drug of interest in cardiology, which is very important for us to understand, is Plavix or clopidogrel. Clopidogrel is given to our patients when they have a stent put in to avoid or correct for a heart attack. And we use it as an antithrombotic, so blood doesn't clot and doesn't fill up that straw that we've put in to keep things open. Now, what's interesting about clopidogrel and Plavix is that it is turned on by CYP2C19. That means it's activated by the body. So you could imagine if CYP2C19 was a poor metabolizer in a patient, that it's just as good as taking candy or eating a sandwich. It's not going to help that patient, right? It's not going to turn it on. And now we're not talking about a side effect of a bellyache or a headache. We're actually talking about a secondary cardiovascular or cerebrovascular event. These are things we can avoid. There are other medications like Brilinta, ticagrelor, prasugrel that can be utilized instead of clopidogrel Plavix. This is showing also that when used preemptively, patients with major depressive disorder respond much, much faster and with improved response versus controls when a pharmacogenomics test is being utilized. And one thing that we want everybody here to understand is that this isn't the future. This is now. There has been a lot of history of challenges and barriers to entry and pushback on pharmacogenomics, but we've reached a tipping point of using pharmacogenomics to improve public health, to improve utilization and ease of use and efficiency for healthcare providers, and to improve patient outcomes. About 20 years ago, the clinical utility of some medications or some gene testing like DPYD was coming into vogue, but more in the academic setting, right? We learned about 5-fluorouracil and capecitabine and DPYD and the potentially fatal outcomes of those medications being used in patients that had those variants. But over time, we've also got improved lab technologies, right? We've got these microarrays that we just spoke about. We've got the targeted assays that we talked about from the open array technologies. We have good implementation models. That is, how do we disseminate this and get this to the most people possible? User acceptance has gone up over the last 8 years, and that means both physicians, healthcare providers, pharmacists, hospital systems, and also patients. And the last piece of the puzzle really was the economic value. It took a long time for studies to be completed to get the economic value. we at Coriell Life Sciences published in March of 2022 on the economic successes of a large program, and we'll talk about that in a minute. I already talked about what we call here in the US the quadruple aim, but that's better outcomes for our patients, improved quality of care for them, lower cost, and also thinking about our physicians, pharmacists, and our surgeons and anesthesiologists that are using these technologies. One thing that I do want everybody in the audience to consider, not knowing what everybody's specialty is here, is that genetics is only one piece of the puzzle. We all need to consider the other risk factors that we in the clinic, pharmacists would be considering anyway. And those are things like drug-drug interactions, contraindications, anticholinergic burden, things like pregnancy and lactation warnings, age-related toxicities, pediatric risks, geriatric risks. All of this needs to be considered together. All right. So if you are just fine to take amitriptyline because that gene has no variants that were detected, we still want to make sure that there's no phenoconversions, that none of the medications that a person is taking, none of the supplements that they're taking, the things that they're eating and drinking get in the way of positive clinical outcomes. So the program that I'd like to talk to you about is called the CorGen Medication Safety Program. The part that we've already talked about is the sample processing using these genetic genotyping technologies. And then we have a clinical decision support tool that is utilized by a pharmacist to review these genetic and non-genetic components for each patient. The beginning of the process, we often use population analytics to see in a larger population who are the right people to test. Now, I'm a firm believer that we should test everybody at some point in time, right? I myself are not on any medications right now, but I happen to be a poor metabolizer at 2C19. So if you see me passed out at a bus stop and have to have a cardiac surgery or put a stent in, please do not give me clopidogrel, all right? Now, what we have to think about is their cost to these programs. And so looking at those people who are going to benefit the most first is often the right way to go. We also look at a component at the very end of the program to understand the improved health and economic outcomes. I'm going to skip these. Suffice it to say that clinical decision support tool is available to make this a lot more efficient, scalable, and fast for our healthcare providers. And what I want to show you here is a case study in the teachers' retirement system of the state of Kentucky. We have tested over 11,000 people already. In the study that we published in March of 2022, we actually were able to evaluate about 5,288 members of this system. What we saw is that even before we tested their genetics, 66% of the participants were on medication that react poorly with DNA. We didn't know what their genetics were already, but what this shows is that, and we've seen this in our studies and others, that 99% of the folks out there, all of us in this room, have a variant that's going to impact a medication that we could eventually take in our lifetime. So again, this is useful both preemptively and also in a reactive manner. If you see that a patient's not reacting well, having side effects, not having the efficacy that they're hoping for, we can use this to streamline our decision-making in the clinic. 3 out of 5 of these members had changes to their medications recommended by a clinical pharmacist. And interestingly, even though these patients have probably been on these medications for a while, we did see that about 87% of them were at high risk of drug-drug interactions. Very quickly in my last 2 minutes here, showing you that the intervention, these were the people who participated in our program, versus those people that were also members of this retirement system that did not participate. They were evenly matched across the board in age and sex and comorbidity, and close to, there was one medication off in the number of medications. But what we saw is prior to the program, there was no savings attributed to the participant or the non-participant group, which is what we would all expect, right? No program, No difference between those two populations. The minute the program starts, we see a cumulative savings here attributable to the program for our participants. There was a per member per month cost avoidance of over $218. Over the number of folks that we tested, as the 5,200 people over 32 months, that was cumulative of $37 million. And what we see is exactly what we would expect in most countries, is that over time, this is prior to the program and our control group, that over time, the costs of healthcare goes up. But what we see in our intervention group is we bent this cost curve down. And even though our participants cost a little more than the control at the beginning, by the end of the 32 months, that's completely reversed. We were able to tie this directly to healthcare resource utilization decreases in hospitalizations of 15%, emergency department visits of almost 7%, and outpatient visits went down over the 32 months, even though over the first year or so, they actually went up because those patients were participating and active participants in their own healthcare. We have since repeated this study in younger patients, active working well population, an international hotel chain saw an ROI of 7.4x. Large manufacturing companies saw a return on their investment of 8.9x. This was a bit higher because we were able to target our enrollment and look at those people who were going to benefit the most. And you can see that that made a good impact. With a laboratory here in the United States, we've got a couple papers published and one in review right now who had good return on the investment. And we're also working with the state of New Hampshire that piloted this and thought they were just going to show it and offer it to the top 40% at risk. They've decided, because of the great results, to open up to all of their employees. I've talked a lot about numbers. I've talked a lot about public health. I've talked a lot about economics, but when it comes down to it, these are all patients. And my role here as Chief Science Officer at Coriell Life Sciences, I'm the lucky one that gets to hear back from patients that their depression has been managed for the first time in 20 years, that their cholesterol is finally dealt with, the muscle aches and pains and the myopathies they were dealing with because of their cholesterol medication has gone away, which means they're out of bed and walking the dog and playing with their grandkids. Behind each one of these testimonials is an actual patient. Hopefully we can all remember that, even though we're talking about these things on a broader scale. So thank you so much. I know we went 2 minutes over, but I really appreciate the opportunity to talk about what to me is a very exciting and actionable clinical program.
Thank you so much, Dr. Schaman. Does anybody have any questions online? You can pop them in the chat box or anything. No? I'm going to move on then.
No questions is either a good thing or a terrible thing. All right, if anybody's got any,
I think it's a good thing. I think you
know.
explained everything really well.
Excellent.
Okay, now we have got Dr. Andrew Macleod, and he is going to be, oh, I can't see. Sorry, give me 1 second, it's not sharing. So like I was saying, next we've got Dr. Andrew Macleod, who is actually with us in person, and he's going to be talking a little bit about pharmacogenomics in a clinical context.
Thanks, Kayla, and thanks to Dr. Coenen and Dr. Schaman. You've both destroyed my speech completely. You've taken every point that I had and moved it on and spoken about it. So I'm going to, there's a little bit of repeating going on here, but I am going to try, can I get this? Where's my numbers? Yeah. Okay. Right. So we've discussed the definition of pharmacogenomics. So I'm not going to go over that. And we've discussed the ADME genes as well. So I'm not going to go over those again. But I want to talk really clinically about why we're doing pharmacogenomics in the first place. And it's very important that it's a meaningful product, that it's cost effective and cost saving, and that it's practical and easy and not going to take too much of our time out of our already busy lives. I noticed that in America, they utilize the pharmacist enormously in this whole pharmacogenomics game. And I'm not sure whether that's going to be a practical solution for us in this country yet. I think that's something we're going to have to look at in the future, but I don't know if we're there yet. So talking about polypharmacy, Jeff spoke about this. We define polypharmacy as the use of multiple drugs to treat a patient. And 1 to 4 drugs is considered normal, 4 to 10 is polypharmacy, and more than 10 drugs at a time is called extensive polypharmacy. This is the study that Jeff was talking about and the cost save that occurs as a result of pharmacogenomically comprehensive medical management. So they look at the pharmacogenomics, they look at the medication, and together they make a decision. And you can see the saving of 37 million Rand and the monthly saving of $219, which is a Rand equivalent of 3,390 Rand, which would cover a main member subscription to just about any medical aid. What is so nice about the algorithm that Coriell gives us is that it looks at drug gene, lifestyle, and demographics of all the patients. And that's what I'm hopefully going to be demonstrating to you today. So my big role is to talk more about the use of pharmacogenomics in a clinical setting, how we can use it quickly, efficiently, and effectively in our practices. As Jeff said, and he's stolen my fire here as well, it's not a standalone answer. We have lots of issues to think about: polypharmacy, disease burdens in Africa being different to elsewhere, access to medicine being different to elsewhere as well. We want to try and go to individualized, personalized medicine. We want to move away from the one-size-fits-all to a patient-centric form of medicine. We're looking at better outcomes for our patients, cost reductions in healthcare, and a utilization away from acute care, which is what Jeff once again showed, that people return to their primary care options and stayed away from the ER and acute care options. Just some big examples that are quite common. The Han Chinese population carries a HLA-B1502 frequency of 10%, and they're at much higher risk than anyone else of developing a Stevens-Johnson syndrome should they be given Tegretol. And the cost of doing a pharmacogenomics test is currently in the States running at around $174, and you could do a comprehensive pharmacogenomics test for that price, and you would be able to identify the people that have the 25% increased risk of having a Stevens-Johnson syndrome by doing that, and that would speak to health economics in a big way as well. I'm not going to go over the 5-fluorouracil thing again, but we have to look in our African ethnicity, African heritage patients, that they have an 8% prevalence of a defective DPYD gene. And in state at the moment, 5-fluorouracil is the one and only chemotherapeutic agent available for chemotherapy in the state sector. Very different in private sector, but that is unfortunately what happens in state. So in terms of how do we utilize this practically, samples can be collected either from blood or from saliva. The forms are filled in through Next Bioscience online. Payment is made online, and 3 to 4 weeks later, reports are generated. So there's two kinds of reports. The first report is a static report. I like to look upon a static report as a photograph in time. This is the medicines you're taking now. Your genes are never going to change, but your age is going to change. Your supplements are going to change, and your habits are going to change. And then you have the live report, where you can change all the variables, change the medication, and as evidence levels rise for drugs, those are going to be added into the algorithm as well. So we're going to have a look at both of those, and I am going to play you a little video on the static report so you understand that. You're going to find in your folder an example of a static report. It's 46 pages long. It's very comprehensive, and I'm just going to play a little video of that for you now.
This short educational video is intended to help clinicians effectively use Next Biosciences PharmaGene Pharmacogenetics report. Firstly, confirm on the opening page the identity of the patient. Then there is an executive summary which is of critical importance in red. This is a list of drugs which are contraindicated genetically in the patient. This is followed by a comprehensive list of the genes analyzed. Here, the activity rather than the variant of the gene is more important, but not essential. Next is the treatment options and associated risks for genetically risky, have drug-drug interactions, and carry anticholinergic burden. This section is specific to the patient. Thereafter, your patient's individual current regimen risk is detailed. Ideally, we would want a score of below 20. The blue lines with the white circles in them indicate black box warnings instituted by the FDA, and these 15 points can be removed from the total score when you see this, but you will see the actual warning listed below. The green means lifestyle interactions, purple means anticholinergic burden, and yellow is drug-drug interactions. Below this is a detail of the black box warning. You can see here that the sleeping pill warns of sleep disorders, and the antidepressant may cause suicidal ideation. Also listed are BS scores in brown for geriatric patients, drug lifestyle and drug-drug interactions. Medication and lifestyle choices are populated into the algorithm from the requisition form. See the sleeping pill may increase Flomax serum levels. Predictive scores from thrombosis follows. Note that a patient who is heterozygous at MTHFR should have folate and homocysteine checked, and folic acid 5 mg daily should be considered. Cardiovascular risk based on ApoE lipoprotein E follows. Patients at high risk need strict control of their lipograms. The best genotype here is 3x3, which is called the wild type. The medication summary in alphabetical order follows, and all drugs known to have pharmacogenetic markers are listed as standard proportions in green, caution to be taken in yellow, and avoid all change in red. This patient, you will see, has a strong contraindication for Tegretol and phenytoin due to the HLA-B status being 15.02 positive. This is Stevens-Johnson syndrome that may be a consequence of using these meds in this patient. This long list is followed by a detailed report, which is really comprehensive and contains medication and its role in your patient's genetic profile. A tick means typical response, yellow means consider an alternative, and red means a drug change is needed. Blue shows that additional information is available. Contact Next Biosciences regarding this. Finally, orange means uncertain response. This is not an essential read unless one of your patient's drugs are deemed caution, or change or score highly in the risk charts. The end of the report has a cutout section for your patient to keep laminated and handy. You can also email the static report to your patients to have available on their mobile devices.
Right. So what I'm going to do now is I'm going to try and demonstrate the live portal to you. If I can get this mouse to work. Where is my mouse? Yeah, please. Onto that thing.
There.
There, yeah. And then we go to projects. And we go into duplicate. Thank you.
Screen sharing is top because the external display is disconnected.
Yeah, that's okay. Yes.
Then reshare.
Yeah, that's fine. That's fine.
It's not sharing. Oh, is it not sharing?
Okay. So what I'm going to do is I'm going to share the pharmacogenomics of 3 patients, and we've got consent from all 3 patients. We're very aware that sharing consent with patients is of patients' genes is something we need to be very careful about. And this is a friend of mine. He's an elderly man, and he presented and he had metastatic stage 4 bladder cancer. And we did his pharmacogenomics before he had his first appointment with his oncologist. And what we found was that MTHFR, he had decreased function at all 3 alleles where they are checked. So now what we're going to do is we're going to go into his, help me. So I'm going to need some help with this because this is a difficult way to do it because of the thing.
Need help with?
And it's just going to the live screen. It's meant to be a plus. A plus. Okay. Plus. Next G does. Okay, thank you. Right. So in this patient, okay, that's right. So I can carry on talking in the meantime. So the standard treatment for stage 4 metastatic bladder cancer is a treatment called MVEC, where on the first day they bring you in and they give you a high dose of methotrexate, and then they bring you back in the following day and they give you cisplatin, epirubicin, and vincristine. That is a standard treatment, and the prescription had already been drawn up because there were no apparent contraindications for this patient. So what I'm going to do is I'm going to go into the live portal now, and I'm going to put in his data, 220190. And because we're doing this via a Zoom call, we have to put in these other things as well, which you wouldn't normally have to put in. So it's just going to take a little bit longer. Okay, 4. Then you have to tick here that you're a licensed healthcare provider, and you're allowed to do that. And you can see this gray bar comes in here, denoting that his genetics have been populated. And then you can see his genetics populated. There's his date of birth. He's an LD gentleman. And I've already populated his lifestyle. So when you populate lifestyle, you just tick whatever box is applicable to your patient. And I've populated his diagnoses here as well. So in his regimen, We've got his vincristine, his cisplatin, and his epirubicin, and he's on valsartan for his blood pressure. And what I'm going to show you is how we put in his methotrexate. Very easy, just like that. It finds it and we put it in, and we can go to his report. How do we get rid of this? Can we get rid of that thing? Okay, that's fine. That'll be great. So we're going to take out the FDA black box warnings. And then what we can see here is that we have a genetic exclaim for the use of methotrexate in this patient. And we have drug-drug interactions. So the vincristine doesn't like the methotrexate, the cisplatin doesn't like the methotrexate, and the methotrexate doesn't like either of the other two either. So we can see here there's a genetic issue here. This patient is like one of those patients that we often see in practice. They have 2 sessions of chemotherapy and they don't make it to the 3rd. And something like this could well be implicated there. May not be, but can be. So we can go to the regimen and we can take out the methotrexate, and the oncologist would then have the option of choosing something else. So I've done a little bit of research, and there is a drug regimen that they use called PADKEV. I'm going to put that in. It is an antibody PADKEV. It's an antibody-based regimen, and we can put that in. And then we can go to his report again. And we can go to his report, take out the FDA black box warnings, and we reduce him to a score of 0 here. So what we can see is that we have minimized this gentleman's risk, and we can say that in all probability, he's going to tolerate his chemotherapy much better than he would be were he getting methotrexate. We can also pull up this thing of side effects and view all his side effects and give this to the patient as well. So then if we go back to the regimen, we can go into what the drugs in the regimen. We can pull out the product education on the patient. This is going to tell the patient all the side effects, everything they should worry about, the interactions and how they would. You can send that off to the patient. So that's what I wanted to tell you about that patient. We're going to go to another patient now. This patient is a young man, still at school, and we are going to be giving him some diagnoses that we would see in young people. He's not on any of these medications, but he has interesting genes. So he's an intermediate metabolizer at CYP2D6. So we can look here. He has his CYP2C19. We spoke about that earlier. You can see he's a star 1, star 2. And at a CYP2D6, he's a star 2, star 4. So those are 2 problems he has there. Also want to show you that he has low activity at COMT and he's ADRA positive. So now what I'm going to do, need to open this up again, please.
That is why I'm standing here now.
Okay. Let's get to stop it. There. Do that. Yeah.
Yeah.
Okay. It's underneath it. Give it a shape. So I'm going to give him 2 diagnoses. I'm going to give him acne and I'm going to give him ADHD. All right. And those are kind of things that all teenagers have, and it would be quite normal for him to suffer with those things. 2, 6. I'm going to put in his key here. A2 M F. Okay. 7 XA. Right. So we have to sign ourselves as a registered healthcare provider and sign in, and let's see. So we've got all his drugs in there. We can look at his demographics. There he is, a young man. His lifestyle changes. He's a young man. So we've given him, let's give him a bit of alcohol, a bit of marijuana. So they all do that these days. And There I've populated his diagnoses in there. What I've done is I put him on Roaccutane, and I'm going to put him on Ritalin. So now you will remember that we saw that he had low COMT activity and he was ADRA positive. Two things that are going to affect his response to Ritalin. So because of his genetics, we can see in his report that he's got a problem with Ritalin. So Ritalin doesn't like alcohol, so that's a lifestyle change. So we're going to say, listen, sorry, you're taking a Ritalin, you don't take any alcohol. And we go back to the report. We can take out the FDA black box warnings, which is for fetal abortion and teratogenesis on Ritalin. So we're not going to worry too much about that in a young man. And we see a big genetic exclaim here. And we can go to the bottom of the report. This is all online. And it says, ADRA positivity may present with increased risk of pharmacotherapeutic failure, the lack of efficacy, and the same thing for his COMT activity. So Ritalin is not the answer for this man's ADHD. So let's try him on Strattera. So we try Strattera, and you can see it just picks it up straight away. Let's put him on 40 milligrams to start with and look at his report. Take out the FDA black box warnings, and he's coming back with another warning. So you will remember that we also said he was an intermediate metabolizer at CYP2C19, which is where Strattera is. So Strattera is not a good choice for him either. So we can go back to his regimen, take out the Strattera, and now we've got to go on to other drugs like Provigil. Provigil, made primarily for narcolepsy, but works very well in ADHD as well. And we can go to his report there, take out the black box warning for that, and you can see that Provigil is going to be a very good thing for him to use. So we're now going to carry on, and we're going to fast forward 30 years for this young man, and we're going to give him a Jeff Schaman cardiac event. So his indication, we can add an indication here. We're going to say atherosclerosis, because that's what caused his heart attack. And we are going to add into his regimen some Plavix, because that is the standard treatment. And if you haven't done genetics, you don't know that the person's not going to be able to benefit from the Plavix as this patient is. But look, it says here, genetic stop, because he's CYP2C19 under metabolizer. So we've got to go back to his regimen and say we don't know what to do. We can go down to alternatives here. And we can go Plavix is out. We want an alternative to Plavix, and it's going to give us all the other platelet aggregation inhibitors that we can use. And what we can see here is we can go to Prasugrel, which is Brilinta, because it's only got those, and we can update the alternatives model here. And there's the Prasugrel, take out the FDA black box warnings, and we've normalized him in terms of his drug response. I'm going to give him one more thing, because we also knew he was a CYP2D6 intermediate metabolizer, and I'm going to give him breast cancer. Very rare in men, but it happens. Breast cancer. Oh no, sorry, I've got to put that under indications, not regimen. Breast cancer. There we go. And in his regimen, we're going to give him tamoxifen after his treatment. Tamoxifen after his treatment. And we're going to see what happens. We're just using his genes here to show us things. So Plavix, we stopped and changed that to Prasugrel. So I'm going to take the Plavix out. And let's go into his report now. And here we are. Take out the black box warnings. And we've got a genetic stop on tamoxifen here as well. So what I'm trying to show is that all stages of your life, you can re-enter the portal. The genes don't change, but you can add drugs, you can take drugs out. And it's such a valuable tool to be able to use in patients. It's quick, it's easy, it's very simple. Genetic stop, so this would then become an area where the oncologist would have to make a decision as to what would need to be done for this patient. And I think we've done very well there. I hope I've been able to show, I'm sorry about all the IT issues, but I hope we've been able to show that how easily it is to manipulate medication and genes with this algorithm. Thank you.
Thank you. And then for everyone who's watching, I just wanted to make this a little bit more actionable. If you are interested in utilizing a PharmaGene test for a patient, yourself, a family member, this is how you'd go about getting that test. You'd go onto our Next Biosciences website, divisions, you'd go to specialists, PharmaGene, then you can go to the ordering process, fill out this TRF, which is digitalized. So there's no need to print and scan. You can fill that out and email that to me at pharmagene@nextbio.co.za. And for everyone who has arrived or has joined in online, I wanted to offer you guys 10% off on a live option of the PharmaGene test that you can offer to a patient, a family member, or you can use it for yourself. Do we have any questions?
We had a hand up from Laura Delan.
Oh, yeah. Okay, perfect. Well, thank you guys all for attending, and online, thank you for hopping in as well. And I hope that you found everything really interesting. So thank you.
Thank you.
Thank you.
Thank you. Bye, everyone.